Good morning, children. Today I am coming with a new content, that is concept of the species. Okay, you know very well, species is the basic unit of classification. This is the first point you should have to remember. The species is the basic unit of classification. Next, who coined the term? John Ray. John Ray is a scientist. John Ray is a scientist. First coined the term species. Coined the term species. Coined the term species. This point you should have to remember. Next one, and his book Historia. He wrote one book. What is that one? Historia. Okay, Generalis. Historia Generalis. Okay, Historia Generalis Plantarum. Historia Generalis Plantarum. How many volumes you know? 3 volumes book. Okay, this book have 1693, in the year 1693 described. Okay, described what he described in that book. Species as a group of, very important, species as a group of morphologically, what you call, morphologically similar organisms. Morphologically similar organisms. Okay, similar organisms arising from common ancestor. Okay, this point you should have to remember. Common ancestor. And up to here, who told? John Ray. Come to Carolus Linnaeus. Okay, Carolus Linnaeus, what he said, in this book, he wrote one book. What is his book name? Systema Naturae. Remember the names, very important book, important neat bits. It is a neat bit. Okay, this is a neat bit. Okay, this is a neat bit. This is the neat bit. Okay, here I am writing two names. I wrote two names. So here what he wrote, considered. Okay, species as a basic unit of classification. Basic unit of classification. Next, species. Okay, species can be defined. Okay, defined as a group of organisms. Group of organisms that have similar morphology, very important, similar morphology and physiology. Okay, these points you should have to remember. And physiology, and can interbreed to produce fertile offsprings. Okay, fertile offsprings. These points you should have to offsprings. Next, this point you should have to remember. In 1859, Charles Darwin Okay, 1859. Charles Darwin. What is his book? Origin of Species. Origin of Species explains the evolutionary, what do you call, evolutionary concepts of evolutionary connection of species. Okay, evolutionary connection of species by the process of natural selection. These points you should have to remember. Okay, children, once listen the what we discussed today, what we are going to discuss, that is a concept of species. So species, S, G, F, O, C, P, K. What is a species? Genus, family, order, class, phylum, kingdom. So species is what is a species? The species is basic unit of classification. First point. Basic unit of classification. John Ray is a scientist, very good and great scientist, coined the term species. It is a neat bit. Okay, John Ray first coined the term species, and John Ray wrote his book. He wrote one book. What's his book name? Historia Generalis Plantarum. Historia Generalis Plantarum have 3 volumes containing book. In that book, he 1693, which year? 1693, he described the species as a group of morphologically, what do you call it, similar organisms arising from, what is that, common ancestor. Very simple. Morphologically similar organisms arising from common ancestors. So, who described John Ray in his book? What is his book name? Historia Generalis Plantarum, having 3 values. Okay, what is a John Ray book? Historia Generalis Plantarum. This is a neat bit. In 1693, he described species as a group of morphological means external characters are similar organisms arising from common ancestors. Their ancestors have common. So such kind of what you called is only called species. Okay, Historia Generalis Plantarum. Next one, the Carolus Linnaeus, the great scientist name Carolus Linnaeus. He is the father of taxonomy, modern taxonomy. He wrote one book. His book name is Systema Naturae. What is his book name? Systema Naturae. This is a neat bit. Systema Naturae. He considered what they considered in the Systema Naturae, Carolus Linnaeus, species as a basic unit of classification. Basic unit of classification. Species can be defined as a group of organisms that have similar morphology and physiology. So morphology, external character, internal physiology, character systems, and can interbreed, means they can reproduce to produce fertile offspring. So fertile offspring, this is a very important. If they can interbreed each other and produce fertile offspring, in the sense that will be considered as species. So species can be defined as a group of organisms that have similar morphology, external features and internal systems, all are similar in the sense, can interbreed. This is a word you should have to remember. Interbreed to produce a fertile offsprings, that is called species. Next one, this is the second neat bit. The third one, in 1859, who is the scientist? Charles Darwin, father of genetics. So Charles Darwin, natural selection, father of natural selection, not genetics. 1859, Charles Darwin created one book. What is this book? Origin of Species. What is this book name is? Origin of Species. He explains the evolutionary connection of species. Evolution means what are the relationship between their ancestors and their present species. Evolutionary, okay, history, developmental changes, evolutionary connection of species by the process of which one, natural selection. Whenever you hear the word Charles Darwin, you should have to remember natural selection. So origin of species explains the evolutionary connection of species by the process of which one, natural selection. So these points you should have to remember. Okay, so first one, what, in 1693 Historia Generalis Plantarum, then Carolus Linnaeus Systema Naturae, and third one, Charles Darwin Origin of Species, explain the evolutionary connection of species by the process of natural selection. So next concept we are going to discuss about, that is about the species. Next, tools, what you called the tools of the study for taxonomy. Tools, okay, observe very carefully. Tools for study, tools for study of taxonomy. These points you should have to, tools for study of taxonomy. Okay, so tools and taxonomical aids may be different for the, so these are different for what, different for what, study of plants and animals. For plants, some tools, study of plants tools are different. For study of animals, the tools are different tools for study of plants and study of animals. Okay, so for example, herbarium, here I am writing herbarium, okay, and botanical garden. Herbarium and botanical garden, use it to, what is the use of this one, study of plant taxonomy. Use it to study of plant taxonomy. It is useful for the study of plant taxonomy. The next one, okay, museums, okay, next one, taxonomical keys, okay, next, geological and marine parks, zoological and marine parks. Okay, these all are museum, taxonomical keys and zoological and marine parks. This is about the full for study about what you called animals. So the important, so this up to here like this, the important components, very very important, okay, components, important components of the taxonomical tools, okay, tools, important components of the taxonomical tools are, number one, field visits. What you call field visits? When a field is there, we can visit and we will find out the information about plants and animals. Next, survey, okay, next, what you call identification, okay, survey and identification. The next one is classification, identification, classification. Next, okay, preservation. Remember, these are all the things we have to do for taxonomic point of view. Preservation and documentation. What you call documentation. These are all the things. Okay, this so helpful for what you called component of the taxonomical tools. Okay, next one we are going to the classical taxonomical tools we are going to discuss. What you called classical taxonomical tools. Classical taxonomical tools we are going to discuss. First one is taxonomical keys. What do you call keys? Very very important. This is also need bit. So keys are based on what do you call comparative analysis. Comparative analysis. Okay, analysis of the similarities. Please remember these names. Comparative analysis of the similarities, number one. And the dissimilarities, you know, same and different. Dissimilarities. Okay, dissimilarities of organisms. Okay, this point you should have to remember. Okay, there are separate keys. Next point, there are separate keys for different taxonomic, very very important, different taxonomic, okay, categories. This is the point you should have to. Similarities: 2 legs, 3 legs, 4 legs. Dissimilarity: 2 legs, 3 legs, 4 legs, 2 legs, like that. That will be called key. Keys are based on what? Comparative analysis. Okay. The next one, museums. What do you call? Museums. Come on, tell you know very well museums, where it all present. Okay. Biological museums. What biological museums have? What these all have? Preserved plants and animals. Preserved plants and animal. Okay, for study reference. Okay, for study reference. This point you should have to remember. Next, for study reference, specimens of both what you call extinct and living organisms can be studied. Living organisms, this point you should have to remember, can be studied. Extinct and living organisms can be studied. These points. Next one, what is that one? Zoological parks. What you called? Zoological parks. These parks you know very well. Zoological parks. So zoological parks where the animals protected, you know very well, zoos. Okay, animals under human care it will be there. And food habits, animals food habits. Okay, food habits and also behavior we can study. Okay, behavior. Okay, these are all the things you should have to remember. Okay students, once you listen, tools for study of taxonomy. There are different tools for study of plants. For plants, study of plants classification study point of view, some tools. For animals, study of animals classification, some tools. Means different tools for plants, different tools for study of animals. That is the first point. Second point, for example, herbarium. What do you call? Herbarium and botanical garden. Herbarium and botanical garden helpful for what? Study of plant taxonomy. Next, museums. First one, taxonomical keys. Zoological parks, marine parks are helpful for what? These all taxonomical tools helpful for the study about animal taxonomy. Next one, the important components of the taxonomical tools are: number one, components, important components, okay, of taxonomical tools are: number one, field visits, survey. First we have to visit the field, then we can survey about that, then identification of one particular or new animal or plant species, then we have to classify that one, then preservation, very useful in the sense we have to preserve that, then we have to give the complete picture in the documentation way. So field visits, survey, identification, classification, preservation, documentation, these 6 steps are the components of taxonomical tools. Next, classical taxonomical tools. The taxonomical keys. First one is key. What is key? Key means not our regular key. Keys are based on comparative. The 2 legs, 3 legs, 2 legs, 4 legs. Okay, like that. So comparative analysis of the similarities and dissimilarities of organisms. There are separate keys. So separate keys for different taxonomic categories. Different categories have different separate keys are there. Next, museum. What is museum? It's a biological museums, preserved plants and animals. So preserved, this word you have to remember. In the museums, what will be there? Preserved plants and animals for study reference. Okay, specimens for study point of view, the dead plants and animals we have to preserve. Okay, so like that, you know very well the animals we can preserve. Okay, so dead body of the humans also we can preserve. You can use, suppose for small invertebrates, formaldehyde we will use, formaldehyde liquid solution we will use to preserve. So specimens of the both extinct and living organisms can be studied. So in the museums what will be there, we can study, we can see, and specimens of what you called both extinct, already extincted, and living organisms can be studied in this place in the museums. The last one is one more important is also there, but you should have to remember the zoological parks. In the zoological parks, all animals are there. They are the protected by humans. Separate where the endangered animals are there, or the endangered conditions, animal situations or habitats. Those all animals the human will be take out and will give protection for those animals, and they will be what you called there, they will study about food, habitat and behavior of those animals, they will be take care. So that is about what you called zoological parks. The last one is there. What is the last one? This is the most important one. So that is a zoological park. The next one is, what is the next one? Marine parks. What I wrote? Marine parks. What are the marine parks? In the name itself, answer is there. Marine means what? So, in protected marine organisms, ocean like that. Marine organisms. Okay, marine organisms are maintained. Marine organisms are maintained in what do you call protected environments. Okay, environments, these points you have to remember. Okay, environments. So that is marine. Last one, a printed, what you call a printed taxonomical, printed taxonomical tools means we have, if you know information means we will print about that particular category of animal or species and we will be. So what are those? First one, in the printed taxonomical tools, identification cards. Okay, if suppose any plant is there, about the plant information we will print and we will paste. Next one, description, we will study about that one, we will keep it there. And the next one, field guides. So these are helpful, these people helpful for explanation about that one, and guides and also manuals. So these all are printed forms. This is a printed, this is a printed. Some guides are there and field guides, some manuals are there, some descriptions are there. These all are helpful for what? Identification cards are there. These all are what you called printed taxonomical tools. What you call printed taxonomical tools. The next category is molecular. What you call molecular taxonomical tools. Molecular taxonomical tools. What are the molecular? I said already, whenever you hear the word molecular, you should have to remember the DNA and RNA. So find this one, the accuracy, okay, and authenticity, remember this name, authenticity is more significant, means compulsory, more significant. So more significant in the molecular tools. So if you want complete correct information, means we have to use the molecular tools. Okay, molecular techniques approaches such as number one, molecular technique DNA, what is called DNA barcoding. So complete information about the purines and pyrimidines of genes. Okay, next one, barcoding. What is the barcoding? Short, remember, short genetic marker, short genetic marker, okay, to identify it as belonging to a particular species, So if you know about the barcoding about that particular species, we can study. Okay, what are the next one? That is about taxonomical tool, classical taxonomical tool we discussed, then molecular we are discussing. The next one, DNA hybridization. What is called DNA? You know very well these words, you should have to remember. DNA hybridization. Okay, these words also you should have to remember. Why hybridization? Okay, degrees measures the degree of genetic similarities. So how many purines and pyrimidines and how those are similar arranged. Okay, similarity, remember this word, between genetic similarities between pools of DNA sequences. Okay, these points also means how many nucleotide base pairs are there in that organism, this organism, and what kind of nucleotide base pairs, how many genes completely, what is the variation that will be give the this DNA hybridization will give that information. The next one, DNA fingerprinting. Okay, these points you should have to fingerprinting. You know very well the DNA should have to. Okay, so first identify from whom one particular fellow, identify an individual. Okay, to identify an individual. So you know very well, murderers, rapists, we can identify them because of DNA fingerprinting. Okay, to identify an individual from a sample, okay, from a sample of DNA by looking at by looking at, very important, by looking at unique patterns. Okay, so unique patterns in their DNA. Suppose two members are there, they're different, what is the difference between them? So that one we will use and we will identify that one. Next, RFLP. What is RFLP? Restriction. Restriction fragment length. Restriction fragment length. Okay, polymorphism. RFLP means restriction fragment length polymorphism. Okay, how the fragment polymorphism, different forms we can. Okay, so what is analysis? RFLP analysis. Okay, what analysis? Difference in difference. Okay, difference in homologous. Homologous means similar. Okay, homologous DNA sequences that can be, okay, that can be detected by the presence of very important, by the presence of fragments of different lengths, different lengths. This point you should have to remember, different lengths. Okay, after digestion of the DNA samples, after digestion of DNA samples. So these all are molecules. DNA level, I said no, molecules. Whenever we hear the word molecules. And also next one is there, PCR, polymerase chain reaction. Okay, like this. These all are molecular the tools. Okay, observe very carefully what we discussed. We are going to come. Marine parks. Extract taxonomical tool is marine parks. In the name itself, answer is there. Marine organisms are maintained in protected environment for study point of view. Next one, printed taxonomical tools. What are the printed taxonomical tools? Identification, description, and field guides and manuals. These all are printed. Means if you know description, if you know, in the sense, we will easily identify which species that is like that. So if you have manual, means complete information about that particular species, we will get it like that. Identification cards, description, field guides, manuals, all are helpful for the complete detailed picture about that particular species. Then what next one? Molecular taxonomical tools. In the name itself, molecular. What is a molecular meaning? Means you know very well, DNA and RNA. So the perfect size, perfect difference, or perfect clarity about the species, or perfect information about without any disturbances. If you want information in the sense, we should have to go for molecular level. Okay, the unique features we will get it by the help of molecular taxonomical tools. What are that one? The accuracy, authenticity is more significant in the means complete picture about the particular species is more significant. Complete picture we will get. Accuracy and authenticity is more significant in the molecular tools. So first one, DNA barcoding, short genetic marker to identify it as belonging to a particular species. So a particular barcoding is there, DNA barcoding, a short genetics marker. What is that one? It is helpful for which species that is. Okay, identify to it as belonging to particular species. So that is a short genetic marker. Okay, what is that one? DNA barcoding. Second one, DNA hybridization. It measures the degree of genetic similarity between pools of DNA segments. Suppose one segment is there, how many base pairs are there? Suppose 10 base pairs are there, in the sense the another one also 10 are there or not. Okay, another stand. So like that, what you called between the two species, or between the measures the degree of genetic similarity, how much similarity is there between the what you call species, between the pools of DNA sequences, the DNA hybridization will be there. There are so many pools are there in the DNA sequences. Okay, in between those pools, the similarities are identified by what you called DNA hybridization. The last one, DNA. Next one is DNA fingerprinting. Suppose any murder case is there. So so many victims are, so many suspects are there. So we have to identify particular murderer. How? By using DNA fingerprinting. Means that unique future of that particular person is useful to guess that fellow by using DNA fingerprinting technology. So to identify an individual from a sample of DNA. So if we get the sample, any blood or any sample, we will get it the DNA by looking at unique patterns in their DNA. So what that fellow particular characters only we can guess, by the help of those characters only we can guess the person. So that is called DNA fingerprinting. What is that one? Individual from a sample of DNA. Next one, RFLP. So in the second year, in the plus two portion, one topic is there about DNA fingerprinting, how to do fingerprinting process and everything given like that. Next year we will study. So next, RFLP. What is RFLP? Restricted fragment length polymorphism. So one particular length of fragment. Analysis. What is the use of RFLP? Analysis differences in homologous DNA sequences that can be detected by the presence of fragments of different length and after digestion of DNA samples. Very simple. Suppose homologous DNA sequences are there. In that homologous sequences, differences are there. Okay, that can be detected by what? Presence of fragments of different lengths. What length is there? One particular in that sequence, how many nucleotide base pairs and what is the length? Another one, how many nucleotide base pairs, what is the length? Comparison. So analysis differences in homologous DNA sequences that can be detected by presence of fragments of different lengths. So length based, after digestion of DNA samples we can get it. That's why it is called restriction fragment. So we will be break the fragment length, and what is the difference between those homologous DNA sequences we will be find out. So that is called restriction fragment length polymorphism. The last one is PCR, polymerase chain reaction. We will get maximum number of what you called DNA sample, DNA what you call fragment. Suppose one DNA fragment to 1 million we can get 1 million samples to study about a particular DNA sequence. So these are all the techniques. What are those? The molecular taxonomical tools. For number 1 is DNA barcoding. Number 2, DNA hybridization. Number 3, DNA fingerprinting. Next, restriction fragment length polymorphism. Next, PCR. These all are helpful to get the complete what you call accuracy. Authenticity is more significant through these molecular tools. Okay, children. Tomorrow we will discuss the automated species identification tools. Automated species identification tools. Tomorrow we will discuss. Thank you, children. Thank you so much. Tomorrow we'll meet.